Yeah, I forget sometimes. But then I can send you guys the recorded lecture if you guys have to miss or you want to watch something back again or whatever. Okay. Everyone got this? I can move on. Okay. To step 3. So we ask the question, and now we kind of want to, try to answer the question. And that's the goal of the 3rd step. It's not the goal, but it's an attempt to answer that question, and we call that a hypothesis. It's an educated guess. It's a guess, right? But it's educated. That's a key aspect. It's not just a random sort of, I think what would be, I don't know why I came up with this, but it's my mind. If I pour mustard on my plant or ketchup, it will grow faster. That was just random. It has no sort of reasoning behind it. What's a better one? If I throw rocks at my plant, it will maximize plant growth. That wouldn't be a suitable hypothesis because it's not really an educated guess. It's more of a random, arbitrary thing. That makes no sense, right? Your hypothesis is grounded in some kind of reason. Maybe water would help my plant grow more. Maybe more sunlight. Maybe the type of fertilizer. So I named a few. So for our example, I like to let the class decide which one do you guys want to test for our hypothesis here. Throwing rocks. Not throwing rocks. We got water, sunlight. Sunlight. I saw sunlight first. Let's do that. So, like I said, the hypothesis is an answer to that question, and it's a guess. And so let's say, right, the question was, how can I maximize plant growth? So I think my hypothesis is sunlight causes or will maximize plant growth. So let's see. So let's say sunlight causes plants to grow. This is our hypothesis. But like I said, we could have said water causes plants to grow. We could have said the type of fertilizer you use, this specific brand of fertilizer causes plants to grow more than others. So that's a hypothesis. It's a guess. We don't know if this is true. We do, actually, but let's pretend we don't. Pretend it was, 300,000 years ago or something. You'll see why I like this example. It makes the scientific method pretty clear, I think. Okay, so before I move on to step 4 of the scientific method, there's 2 criteria for a hypothesis that are necessary for the hypothesis to be valid or correct. So if I go back, what is a hypothesis? So we sort of defined it as this guess, right? But it's educated, like there's some sort of thought going into it. Well, I think that sunlight contributes. And so there's two criteria necessary for a good hypothesis. Let's write that down. And I'll go back. Two criteria necessary for a hypothesis. Oh, got someone in the waiting room, admit. Oh, let me check my email really quick just in case. Looks like most people are here though. Okay, Crystal got in, cool. All right. So the two criteria that are required for this hypothesis, it must be testable. We have to be able to test our hypothesis, okay? So that's the first criterion. You can't have a good hypothesis that you can't actually see if it's correct or true, right? So that's our first criterion. And the second is it must be falsifiable. Falsifiable. Meaning it has to be able to be rejected. It can't automatically be correct or true. It seems very obvious, these sort of criteria, but it's in the textbook and I have to teach you. And it makes sense, right? You don't want to, good hypothesis isn't something that can't be proven false, right? Otherwise, why test it? So maybe sunlight doesn't cause plants to grow, right? Of course, like I said, we know the truth, but yeah. Any questions so far? Okay, cool. So I'm actually going to tackle two things on this slide that I'm on: hypothesis and these criteria, and also independent and dependent variables. So I will write that in a moment. Oh, cool. Okay. Now, I'm wondering if I should erase and write this. I think I will. So I'm going to write this here. All right, so every hypothesis has what we call an independent variable. Oh, I'm going to do it in blue. Independent variable. And we actually abbreviate that IV. Put that in parentheses. I don't like this blue, it's running out. Backups. Okay. Every hypothesis has an independent variable and a dependent variable, which we abbreviate DV. Oh, crap. So we have an IV and we have a DV. Okay, so what are these? What is an independent variable? What is a dependent variable? So I've actually looked into the definitions before, and they weren't too great. I didn't really like them. So I kind of talk through it and I kind of explain it in my own words. But I did actually recently, I taught somewhere that defined it sort of well. So I'll mention that. And it was in the lab, and the scientific method lab, and it defined it. I was like, all right, that's decent. So the independent variable is the variable that's manipulated by the researcher. That's what the lab says. It is the variable that we choose to test, okay? We chose to test the independent variable. The dependent variable, that lab defines it as a measured effect, a measured change, something that's measured, okay? I like to define it as the thing that is, yes, measured, but an effect. Something that you can test and measure that is an effect of the independent variable. So the independent variable, again, is something we choose that is going to cause a measured effect that you can, well, it actually depends on that independent variable that we chose. And I also like to mention that it's something that we have no control over as researchers. That's why it's dependent on what we chose as the independent variable. Now I'm gonna, I think it'll make a lot more sense when we look at some examples, namely this one first. Okay. So with that brief explanation, yes, what do you think is the independent variable in our hypothesis? Sunlight causes plants to grow. The sunlight, very good, cool. I feel like I've explained that better in the past, but yes, we chose sunlight. But we could have chosen water. We could have chosen fertilizer type. We could have chosen type of plant. This type of plant might not grow as much. That sort of thing. Now, what's our dependent variable in this example? The growth, the plant growth. So, nice, great. The reason that is because the amount of sunlight, the sunlight itself, we chose that. We can manipulate that. We can do less sunlight, more sunlight. That's up to us. But the plant, its growth depends on that sunlight that we chose. Maybe if we chose water, the growth of the plant would be more than sunlight. Maybe water causes it to grow more. That's out of our control. We have no control over how the plant decides that, right? So it is a direct measured effect that depends on what we chose for that independent variable. Looks like you guys got it. Okay. Takes me a minute to get back into lecturing mode, even though I'm talking to my wall. All right. Let's look at some examples and practice this. Okay, so for each of these hypotheses, define or identify the independent variable and the dependent variable. Okay, they are all hypotheses. So, drinking a glass of red wine once a week prevents heart disease. So what is the independent variable here? What is the thing that we chose to test? Yeah, the wine, exactly. Awesome. We could have chosen exercise, right? Exercise 3 times a week. We can decide the frequency. That's up to us. Prevents heart disease. Well, we can't choose that, right? So what's our dependent variable? Right, heart disease, whether you get heart disease or not. It depends, right? Whether you get heart disease depends on our independent variable that we chose, or if the red wine will prevent it, cause that prevention, okay? Let's look at another example. Students who study 6 hours a week earn higher exam scores. So what's our dependent variable? Exam scores. Perfect. Perfect. All right. Independent variable. Nice. Studying 6 hours. Exactly. Could have chosen sleep, amount of sleep. Could have chosen whether you come to office hours, whether you do the study questions, the extra credit study questions, earn higher exam scores. What will cause that effect, right? Awesome. Cholesterol levels are lower in women who eat 2 bowls of Cheerios a day. What is our independent variable? Nice. Cheerios. Yes. Oops. Why can't I? Sorry, I screwed up. Okay, let me do that again. Please be fixed. Darn it. Can I move this? Oh, I can move this. There we go. Sorry about that. All right. Cheerios, perfect. Okay. Dependent variable. Cholesterol, you were ready. Awesome. Last one, plants. that are grown under red light grow faster than plants grown under natural sunlight. Okay, what would be our independent variable here? So the type of light, is it the red light or the natural light? So we were testing type of light that we are exposing our plant to. And then our dependent variable would be what? Growth. Growth, perfect. Plant growth depends on the light we chose. Awesome. Okay. How are we doing on time? Okay, we're good. So I want to show you a funny video clip. It's more, you know, it's it's just an interesting thing that exists and it's real. But oh, did I do that first? Okay, never mind. We'll do the clip in a minute. So after the hypothesis, the next step of the scientific method, okay, is the experiment. And I pretty much wrote everything here, actually. So I'll write it here anyway. So we have our hypothesis, and now we want to test our hypothesis by doing an experiment. And that's the fourth step of the scientific method. The experiment. Okay, so we're going to conduct our experiment with proper technique controls we're going to discuss in a moment. You're going to collect your data. So in fact, I got to update this slide to say that the next step in the scientific method, so if I go back here, after we perform the experiment, we're going to be generating data. We're going to be collecting data from this experiment. And the fifth step is to analyze the data. Let's look at it. What does it tell us? So analysis. And then, like I said, some textbooks will have a results section, but I just go straight to the conclusion. What does our data tell us? So the sixth step after we analyze is our conclusion. So I'm gonna, if people are writing what's on the slide, I'll just go back. Again, this is available on Canvas. Also, if you see me looking at my phone, it's actually where I have my notes. I'm not texting people or on Instagram or anything, I promise. All right. Okay, so let's take a look at this video, this example of a scientific experiment that was done. That's kind of funny. Okay. So I should have it up. I do. All right. So let me know if you guys can see and hear this.
Are you looking for love and failing to find it? In the past, retailers have sold love potions that claim to attract the opposite sex, even though none of it's been proven to work. So we were curious about the new batch of potions with the secret ingredient pheromones. They're in some of the new Christmas fragrances, but do they work? Bill Ritter did a little experiment to see if they're too good to be true.
They are identical twins, 23-year-olds Sarah and Bridget. They're getting ready to go out to answer a question. Can you make yourself more attractive just by using an over-the-counter chemical that's peddled as something of a love potion? Another set of identical twins is also helping us, 25-year-olds Dave and Paul. They check their hair and put on some scent that we've given them. Both sets of twins meet up later and head to their final destination, a bar in downtown New York City. The twins have volunteered to participate in a 2020 experiment.
Jim, nice to meet you. I'm Sarah.
A speed dating experiment to help us find out if these chemicals called pheromones actually work.
That's where I'm at right now. Exactly.
Pheromones are natural and odorless. Many animals secrete them to attract mates. But pheromones are also synthesized and marketed for humans. And if you believe the package label, they enhance sexual attractiveness. Now, pheromones have been around for years, but now new studies and new claims make pheromones hotter than ever.
I think our product is the only product that has any proof behind its claims.
Biologist Winifred Cutler is perhaps the nation's leading pheromone champion. She claims her pheromones work for 75% of the people who use them. The price for 1/6 of an ounce of her pheromones, sold through a company she calls the Athena Institute, is $100.
Our pheromones are sexual attractants. They do make the wearers more sexually attractive. That's very clear.
And that's where our identical twins come in, because with them, the whole package is the same. What happens to one should happen to the other. So what do you expect to happen tonight?
In the back of your head, you're thinking, yeah, this is, this stuff really works. Then,
Right, I'm wearing the secret formula.
Exactly. Like nobody else has this.
Right.
For
our experiment, we gave all the twins a scent, but only one twin in each set got the scent with Dr. Cutler's pheromones mixed in. And we didn't tell them who had this secret weapon.
The gentleman, when I ring this bell every 5 minutes, you're going to rotate one table up. I'm Sarah.
David.
And I'm Bridget.
10 different dates, each one lasting just 5 minutes.
Okay, gentlemen, it's time to rotate.
Would the pheromones make a difference?
What about yourself, Aaron?
Gentlemen, one, please.
Nice to meet you.
Born in Queens.
Well, it's good to meet you.
This is your final date.
At the end of the night, we tallied the results. Of the 10 dates, 9 men wanted to see Sarah again, but Bridget attracted just 5. As for the male twins, Dave got 10 women interested in him. Paul had only 6. So who was wearing the pheromones? Sarah and Dave, the two who scored better. Oh yeah, I know what you're thinking. Why would attractive 20-somethings even need pheromones? After all, it's those of us who are no longer in our 20s who could probably use a little help.
Yeah, it's that's so weird what he says at the end. I'm like, okay. All right. So let me go back to the PowerPoint here. All right. So yeah, this is a real company, Athena Pheromones, and they claim it works. I'm sure some of you are skeptical after seeing some of the data that they're collecting there. Even though it looked like it worked, we're going to talk about maybe why that's not the most reliable experiment that they did. So anyway, I thought, it's a good example to talk about with the next learning concept. Oh, if we have time at the end, we can discuss these questions, but I'm gonna, like I said, it's, I mentioned not the best experiment. Like, does it really work? We'll talk about that. But for now, I want to talk about what is our independent variable here. So let's start with that. What is our independent variable for this experiment? Correct, exactly. The pheromones, the perfume one. Yeah, what they sprayed, it has those pheromone chemicals. And we're gonna see if that causes what our dependent variable is. So what is that? Our dependent variable depends on these pheromones. Yeah, attraction, yeah, sexual attraction, or the number of dates, right? Is it gonna depend on if they're wearing the pheromones or not? Can we make that reliable conclusion? And so the next question I asked, what were the controls? Now we haven't talked about that yet. So let's talk about that. Controlling an experiment. Okay, 1 second. Okay. Sorry, sometimes I write little notes to myself, like if I want to reorder slides in the future or something. So sometimes I take little pauses here. Okay. Okay. Controlling an experiment. So what were the controls? Let's learn what that means. Okay. So a control group is not given the independent variable, okay? That's our control, or control group. They may be given a placebo. So what's a placebo? A placebo is a fake treatment, right? And we saw that actually. We saw that in the experiment that the twins, right, take the two guys, one of them, well, they both, right, so both of them, like Christine says, both of them got bottles, right? Not just one of them, but they both got bottles. But only one of them had the pheromones in it. But why did they get both? Why did one get a placebo, just filled with water with none of the pheromones? We call that a blind experiment. So I'm going to write that here. It's a blind experiment. This is something that is really necessary and much more common with human studies than if you're working with cells in a lab or mice, right? But this is a blind experiment. And what this means is that the participants don't know if they're in the experimental group or the control group. So let's, I'll mention that, well, first I'll mention our independent variable was what again? The pheromones, exactly. So our control group, our control twin, the twin, yeah, does not have the pheromones. We need controls. We need a control group in every scientific experiment. We're going to talk about the criteria soon about for what makes good science. Funny story, because of COVID going on, my dad and I talk a lot. He's an urgent care doctor, and I talk to him a lot about his ideas about all this. And we don't think the scientific method has been properly executed. To the disagreement to a lot, but I won't get into it, but it's fascinating to be critical and to analyze deeply. And every scientific experiment, you need a control. You need a control group where you take that independent variable, in this case, the pheromones, out. And we'll talk about why. Because the reason why I like to refer back to the plant example we were doing. So yeah, I'll mention it now. Perfect time. In our plant experiment, our experimental group was the group of plants that received sunlight. We're going to see, does sunlight cause these plants to grow, right? So I have plants, I planted them, and I give them sunlight. What is my control group going to be for that example, for the plant example that I love? Am I giving my plants sunlight? So placebo is not that important because plants don't really know. So really quick, the reason we give a placebo in human experiments, so this is, sorry, I should have mentioned this before. This is important. Anyone heard of the placebo effect? Like I said, this is more of a human study type thing where you have blind experiments. If one of the twins knew that they had the pheromones, it gives a bias because that twin might be like, oh, I have the pheromones. I don't even need to try. Or maybe they'll be like, yeah, I got the pheromones. I'm gonna get as much as I can. It might influence how they act. Versus the other one, if they knew they didn't get the pheromones, that they got the placebo, they'd be like, maybe I'll try harder because I don't have them, right? So it's really a human study type thing. If you go into psychology or drug development, they use placebos more. But plants don't know. Plants won't be like, oh, I'm not getting sunlight. I'm just gonna not grow as much. No, they don't. As far as I know, they don't have a mind of their own. So plants, the control group will get artificial light or no light. You take that independent variable out, okay? This is the reason you need a control. I cannot tell you that my hypothesis is true or my data support these hypotheses if I do an experiment where my plants, I give them sunlight and they grow. And I say, hey, I'm right. Sunlight causes the plants to grow. My PI or my boss might be like, well, where's your control group? And if I say I don't have one, he's like, well, I don't believe you then. What if they just grow without sunlight? Take the sunlight away. Do they grow? That's why you need control. You got to take that sunlight away and show, hey, they don't grow. Therefore, the sunlight is causing the plant growth. Because when I took it away, the plants did not grow as nearly as much as they did with the sunlight. So we can make that conclusion. But like I said, without that control group, you see those plants grow with sunlight. I could say, well, maybe they grew that way anyway, without sunlight. How can you say the sunlight caused it? You got to take it away and show that they didn't grow. And in this example, you would not give pheromones to one of the participants and say, hey, this person with the pheromones got way more dates than this person without them. Therefore, the pheromones have an effect. My hypothesis is supported by my data. Does that make sense? Gotta have control, or I won't believe you. No scientist will. I got in trouble in grad school because I was too lazy to do controls and I had to spend an extra like 3 months doing it. Okay, so I learned my lesson. Let's talk about another concept here called double blind. There it is. Sana, what's double blind? Perfect, you're ahead of me. Here we go. So a double blind experiment So a so-called double blind is when not only are the participants unaware of who received the independent variable, but the people conducting the experiment, the researchers, the scientists, don't know either. And we'll talk about why that is. So both the researchers and participants in the study are blind. Like I said, I like to get you guys involved because you're staring at a screen. I don't want, I want to keep it exciting and interactive. So yes, the researcher is the one doing the experiment. So what will happen in a double blind experiment is the researcher will actually give instructions, give all the protocol materials to a third party offsite, a clinical trial site, whatever it is, who will perform the experiment for the researcher. So he has no idea, he or she has no idea who's receiving the drug and who isn't. So let me ask you guys, why? Why is this a good thing? Why is it important? Why might it be important that the researcher has no idea who got it too? Bias, cheating, absolutely. What else? Bias, perfect. That's exactly it. I like to say fame, corruption, money, because maybe they'll know who got the drug and say, oh, well, I need another publication so my grant gets funded. So maybe I'll skew the data for this participant a little bit because I know they got the drug. And then I can show the grant, the government or the NIH, whoever's funding me, that it's working. Give me more money. It's a little extreme, but really it's this bias. It's to have the most objective data. So even when the researchers you know, it could be unknowingly writing the conclusions, will say or be very honest and not have a tendency to exaggerate the results, like, oh, this person, you know, or the data collected at least. But that's the idea, absolutely. Okay. All right. The next thing I want to discuss with you guys, actually, I'll take a pause here. Does anyone have questions? We're doing pretty well on time, last I checked. Yeah. It is hot. All the fires and stuff, man, crazy. Okay, what about an experiment that can't have a control? That's a good question. I don't know if I can think of one. But sometimes it's hard to, I'll admit, sometimes it's hard to make a control like convince you enough. But I can't think of one on the top of my head. How do the two criteria apply to hypotheses about space? Ooh, we're going deep. So itself, I'm not an astrophysicist, but you know, it's very interesting is from what I've like seen of that kind of stuff, they are very convinced that their data is testable, or their hypotheses rather are testable, and their data is very accurate and convincing in that regard. You know, they have such amazing technology these days when it comes to that kind of stuff. How the height affects the force of ball. Oh. James, you got me. That's a tough one. Maybe James, like taking the ball. So James asked about if there are experiments where you can't really have control. So how height affects the force of ball has landing on the ground. I would take away the height, maybe, and maybe drop the ball from a much lower height, or maybe, that's my best guess. That's a physics one, so I'm not as, that's a tough one. You got me. Most experiments you can have a control, and you'll see any published paper that's peer reviewed, you're gonna see a control group there. Reading Sana's question, what we manipulate. Yes, that's exactly correct. That is what you need to know. I would also know, if I give you an example, maybe identify the independent variable, dependent variable, but that's exactly, so actually, Sana, I misread. So what you manipulate is not the dependent variable. That's the independent variable. Remember, the researcher, we chose sunlight, could have chosen water or soil type, fertilizer. But the growth of the plant depends on what we chose. That's why the dependent variable is the measured effect of what we chose that causes that effect. Okay, cool. Great questions. Okay, another very important concept, control variables. Sharing, right? Okay. Okay. Control variables, conditions that are the same in both the experimental group and the control group. We call these control variables. Okay, so what does that mean? Let's look at our speed dating example and then the plant example. So with the speed dating, one controlled variable that we kept between, so remember the experimental group was the one who received the pheromones, control group got the placebo, the controlled twin. Now, so we actually controlled for their physical appearance by choosing twins. They were the same in both the one who received the placebo and the one who did not. So we controlled for physical appearance in that example. So we would call that a controlled variable. Because we use twins, right? Oh, let me, okay. We also controlled the environment, speed dating. Each date lasted around 5 minutes, right? This was the environment. Now I'm going to talk about why this is important in a moment. Underneath, we'll list some control variables for our plant experiment, so I'll just do that for now. Okay, now the reason we want to keep conditions to be the same in both our controlled and experimental groups. The reason is because we don't want anything to influence our results, anything at all, other than that independent variable. If my hypothesis is that the pheromones cause or increase attractiveness, then both my control and experimental group, nothing in those groups should influence that possibility besides the pheromones, right? So physical appearance, we can't have one look different than the other. That might influence the attractiveness, right? The amount of time on the date. What if one of the groups, they spent like 30 minutes and got into this deep conversation and they connected so much, and for every date, they're gonna get more. So we kept it at 5 at each date. The only difference, according to the researchers, right, was that the experimental group got the pheromones and the control did not. And in an ideal experiment, you only want, the only difference between your controlled group and your experimental group is that independent variable, whether you have it or not. You want to keep everything else the same. Now, I love to bring up the plant example again with this. So we use sunlight. What are some things that we want to keep the same in the control group and the experimental group? We don't want anything else influencing plant growth, right? Soil. Appearance is, I would say, pot size, sure. Watering, absolutely. Water, that's a great one. If I have my plants that receive the sunlight, my experimental group, and then plants that don't receive sunlight, if I give my plants that don't receive sunlight more water than the ones that receive sunlight, they might grow the same amount. And then you can't say sunlight causes plant growth anymore. Does that make sense? The only way you can make this conclusion that sunlight causes plant growth is if you keep everything the same between the two groups besides sunlight. What if my soil type had more nutrients in it in my control group? Well, it might grow more. Yeah. Does that make sense? Let me see questions here. Yeah, the writing on the board is related to the control variable. Same kind, same environment, same plot, absolutely. Type of plant even. What if my experimental group that receives sunlight, those plants don't grow that much anyway, but then I used a plant that doesn't really need that much sunlight, a type, right? What if it's like a Seattle plant, right? Cloudy a lot, and it grows anyway a ton. Then you can't make, your data is gonna be screwed up. You might get the same results on both sunlight and you take away the sunlight and it's still growing. And then you're like, wait, sunlight doesn't cause plant growth, I guess. No, your methodology, you gotta have those controlled variables. Okay, so let's list some. Water, type of soil, plant type, etc. You want to try and keep everything the same so that it is a very direct and accurate measure of what the sunlight's doing. I can make a reliable claim that, hey, this sunlight causes this much growth. And it's purely due to the sunlight because they both receive the same amount of water, they both have the same soil, both the same type of plant, same fertilizer. It's hot. Okay. Oh, recording, I email after every lecture the recording. About an hour or so after it sends me the recording and I email it to you guys. Okay, so 7 minutes left. So there's one more thing I want to cover, which I was alluding to earlier. How do we know if it's reliable? I mentioned some things, right? The controls, we got to have a control, and that's going to be part of this. Okay, so there's four criteria for how we can tell if if I look at a scientific study, this is what I'm looking for, okay, in order for me to trust it. It's repeatable, meaning do other scientists come to the same conclusion if you repeat the experiment? So repeatable. Do other scientists come to the same conclusions? Second thing, what we've been talking about for the past 30 or so minutes, proper controls. So I'll say methodology. We we do that plant experiment, am I giving the same amount of water to my plants? Am I using the same type of plant? Want that to be in order. Another one that I don't think we'll have time to discuss it too much, but in the dating example, I'm not going to believe it that much because the sample size, the amount of participants that they used was very small. They used 2 pairs of twins. So we call that having a large sample size. That's going to increase the statistical reliability. Now, if they had, 100 twins that did it, and then they saw that 90 of them, the pheromones, it resulted in more dates, I believe it a little more. But usually with these kinds of studies, to make that kind of reliable conclusion, you're going to need like thousands. And like clinical trials, things that are FDA approved, they take so long because they use hundreds and hundreds of people to make sure this drug doesn't cause disastrous side effects, things like that, which a lot of them do anyway for a small portion. But you got to test a lot of people to determine that. Okay, the last thing, if your study's published in a peer-reviewed, reputable scientific journal, usually that's pretty good indication that it's reliable. Yeah. So peer-reviewed means that other experts in your field, like say you publish in a marine biology journal, and you found something about corals, other marine biologists who are experts on corals look at your data, look at your findings, and see how you did it, and they give you feedback, and they correct you, or they send it back to you, and then you revise it, etc. So this is called published in a peer-reviewed journal.